Radiopharmaceuticals, inorganic. This topic is a part of the subjects pharmaceutical inorganic chemistry and radiochemistry. Content of this topic includes radioactivity, measurement of radioactivity, properties of alpha, beta, gamma radiations, half-life, handling and storage, radiopharmaceuticals, applications. Radioactive substance is a compound that has radioactivity. Radioactive substance is unstable and spontaneously decomposes and emits radiation, which is not influenced by temperature, pressure, or catalysis. Radioactivity is the emission of ionizing radiation or particles caused by the spontaneous disintegration of atomic nuclei. Units of radioactivity. First unit is Curie. Abbreviation is CI. The quantity of material that undergoes the same level of disintegration in unit time as 1 gram of radium, and that is equal to 3.7 plus or minus 10 raised to 10 disintegrations per second. Units like millicurie or microcurie is also used. Second unit is Röntgen. Abbreviation is capital R. It is the unit of exposure to gamma radiation. 1 Röntgen is equal to 2.58 into 10 raised to minus 4 coulombs per kg. Radiation absorbed dose. Abbreviation is RAD. Unit of adsorbed dose of any ionizing radiation. 1 rad is 100 erg per gram. 1 erg is 1 dyne centimeter. 1 rad is 10 raised to minus 2 joule per kg. Pharmaceuticals are described in unit of rad. The next unit of radioactivity is radiation equivalent man. Abbreviation is REM. It is the unit of human exposure. Takes into account the biological effectiveness of different types of radiation. Explosion rate constant. It is the dose rate in Röntgen per hour at 1 meter distance from 1 Curie source. Relative biological effectiveness. Abbreviation is RBE. It expresses the relative effects of radiation, alpha, beta, and gamma, on the biological system. RB is the ratio of the absorbed dose of a reference radiation, X-rays or cobalt-60 gamma rays, to the absorbed dose of a test radiation required to produce the same specific degree of biological effect. The measurement of radioactivity involves quantifying the rate of radioactive decay and its effects using specific units and specialized instruments. First instrument is ionization chambers. It is available in various sizes and shapes. Consists of a chamber filled with gas. Ionization chamber is connected with 2 electrodes kept at different electrical potentials and also connected with a measuring device to indicate the flow of electrical current. Radioactive material emits radiation. This radiation Ionize the gas molecule. An ionized gas molecule changes electrical current. The change in current reveals the radioactivity of the material. Second instrument is proportional counter. It is modified ionization chamber in which ionization of gas and removal of electrons causes production of more free electrons. That is, an amplification of the current. The voltage ranges over which the amplification occurs is called the proportional region, and the current working in this region is called proportional counter. Most popular counter of radioactivity is Geiger-Muller Counter. Geiger-Muller Counter is a device for measuring radioactivity by detecting and counting ionizing particles. Construction of GM counter. It contains cylindrical cathode and wire anode. Space is filled with ionizing gas and quenching agents, for example, bromine, chlorine, and ethyl alcohol, ethyl formate, as organic quenchers. For solid samples, the end window type GM counter, window made up of an aluminum alloy, mica, or glass bubble. For thin glass-walled counter, glass-walled with 20 to 40 milligrams per square centimeter. To count the medium and high-energy beta particles, it is coated inside with graphite that acts as a cathode. For liquid samples, have capacity with 10 centimeter cube annular pace and capacity with 5 centimeter cube annular pace. For gas samples, gas sample is introduced along with counting gas. Operation or working of GM counter. When an ionizing radiation enters the counter, primary ionization takes place, and a few ions are produced. These ions are multiplied by further collisions. Thus, a bulk of electrons is produced in a short interval of time. This bulk, on reaching the anode, generates a current pulse, is counted by the instrument. Pulse rate versus applied voltage, Geiger characteristic curve. Increasing external voltage from 0, counting will start from particular voltage, called the starting voltage. As the voltage increases from starting voltage, counting rate increases rapidly. This voltage is called the threshold. If voltage increases from threshold, counting rate become nearly constant. This voltage range is called plateau. An operating voltage is selected within the region of plateau. If voltage increases from plateau, second time counting rate is rapidly increases. This voltage range is called the discharge region. Why are quenchers used in GM counter? After removal of an electron, positive argon ions reach to the cathode to gain electrons and become excited state neutral argon atoms. These excited atoms return to the normal state with the emission of photons, and these photons cause avalanches and hence extra pulses. These unwanted pulses are avoided by the use of quenchers, such as bromine or ethanol. in the tube. When positive argon ions move towards the cathode, they collide many times with the quencher molecules and transfer their charge to them, and argon become neutral. Thus, only quencher ions reach to cathode, where they gain electrons to become neutral molecules in an excited state. However, the excited quencher molecules lose their excitation energy by dissociation into normal atoms, and not by photon emission. Thus, no spurious pulses are produced. Scintillation counters work by converting the energy of the radiation into light by scintillator, which is then detected by a photomultiplier tube. Common scintillator materials include sodium iodide, cesium iodide, and plastic scintillators. Properties of radiations. First is alpha rays. Alpha ray, made up of 2 proton and 2 neutron similar to helium nuclei, has least penetrating power and has positive charge, +2. It is deflected by a strong imagistic film, emitted by radioactive heavy metals. These particles produced 30,000 ion pairs per centimeter path. Example of alpha emission from uranium. After alpha emission, the resulting daughter nuclei have 2 positive charges, less than original nucleus. Hence it gives an element, thorium, having 2 atomic number, less than the original nucleus uranium. Second type of radiation is beta rays. These rays are electron-like particles emitted from the nucleus, has more penetrating power than alpha. Beta rays, able to pass through an aluminum foil of a few millimeter and show less ionizing power than alpha. There is 2 types of beta rays: positron, that is, positive beta particle, and negatron, that is, negative beta particle. For example, carbon-14 emits beta particle and converts to nitrogen. The net effect of beta emission on a nucleus is that a neutron is converted to a proton. Emission does not alter the mass, but alters atomic number, converting a radioactive element, like carbon, to the next higher atomic number atom, as nitrogen. Positrons are not very common, as they are short-lived particles. Next is gamma rays. Gamma rays have electromagnetic wave properties rather than particle properties, having the speed of light, more penetrating power than alpha, and beta, no mass, no charge, poor ionizing power than alpha and beta. Iron-59 emits beta and gamma radiations and spontaneously converts in cobalt-59. Gamma rays are produced during radioactive decay, along with beta particles. Gamma emission does not alter the nuclear energy level, and no elemental changes, unless emission of other radiations within the same material. Half-life. The half-life of a radioactive element is the time needed for half of the material to decay. Equation is T 1/2 is equal to 0.693. Divide by lambda, where lambda is disintegration constant in unit per second. Handling and storage of radiopharmaceuticals. Handling with great care. Do not touch radioactive material. Materials must be carried in trays having a layer of tissue paper. Use rubber gloves, protective clothing. Use inactive toward radiation glassware. Waste material should be stored until it is no more radioactive. Storage. Stored in rooms, away from roads, and working areas. Provide shielding with thick glass or metals like lead. Stored area must be checked for radiation leakage. Container labeling with shielding. Disposal must be under sufficient monitoring. Regulatory guidelines for safety. International Atomic Energy Agency, radiation protection and safety in medicinal uses. IAEA, WHO, good manufacturing practices for radiopharmaceutical products. Provides minimum GMP requirements tailored to the unique characteristics of radiopharmaceuticals, such as short half-lives and small batch sizes. International Commission on Radiological Protection, guidance on radiation protection, including dose limits. International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, guidelines on quality, safety, and efficacy, E-Series, for pharmaceuticals globally. U.S. Food and Drug Administration, guidance on non-clinical evaluation, clinical trials. Radiopharmaceuticals. Radiopharmaceuticals are unique medicinal formulations containing radioisotopes that are used in major clinical areas for diagnosis and/or therapy. General applications, medical uses, in 4 different ways: radiation source, in therapy, radioactive tracers, for diagnostic purposes. Research and sterilization of medical products. Sodium iodide. This contains iodine 131, available as oral solution, intravenous, or capsule form. Properties. It is clear colorless, darken upon standing due to radiation. Preparation. Tellurium 130, on bombardment of neutron, emits gamma and converts in tellurium 131, then after spontaneous emission of beta rays forms iodine 131. For injections, preparation, preservative, benzyl alcohol is added, reducing agent, sodium thiosulfate is added, pH maintained to 7.5 to 9.0. Storage. Containers used are previously treated or washed with 0.8% sodium bisulfide and 0.25% sodium iodide, and last, with water to prevent the adsorption of material. Proper labeling with expiry Use in treatment of hyperthyroidism. Iodine 131 selectively concentrates in the thyroid gland, where decomposes to xenon 131 with emission of beta and gamma rays. These beta particles lead to highly localized destruction of thyroid follicles. Use as diagnostic aid, study of the thyroid gland, tumor location. If uptake of iodine is less than 10%, the patient has hypothyroidism. If uptake of iodine is over 50%, the patient has hyperthyroidism. Patient drinks a sample with a small dose of radioactive iodine 131. Thyroid uses iodine to make thyroxine. After 2 hours, uptake of iodine is measured by scanning with a radiation detector. Asymmetric scan indicates disease. Rose BENGAL, labeled with iodine 131. It is prepared by refluxing rose BENGAL acid with radioactive iodine. Product is precipitated with acid, giving radioiodinated rose BENGAL acid. Neutralization with sodium hydroxide gives the sodium salt of the drug. Used to study of liver, evaluating the jaundice patient. Ferric citrate with radioactive iron 59. It is prepared by neutron activation of iron 58. Formulation contains preservative, benzyl alcohol is added, and antioxidant, ascorbic acid, is added. Properties: iron 59 emits beta and gamma rays and converts to cobalt 59. Use diagnostic studies of iron absorption, plasma clearance, metabolism, and RBC formation. Investigation of hematological disorder. Sodium phosphate, P32. Prepared by neutron activation of sulfur. Properties: clear, colorless, darken upon standing, due to radiation. pH maintained at 5.0 to 6.0. It is existing in the form of monobasic and dibasic phosphates mixture. It is decomposed to sulfur 32 on emitting beta radiation. Uses: diagnosis and treatment of neoplastic diseases. Phosphate is used in cell metabolism, and cancer cells have the highest turnover of phosphate. The accumulation of phosphate in the cancer region is identified by labeling. Study of RBC and WBC associated diseases. Treatment of polycythemia, that is, increased level of RBC. Treatment of myelocytic leukemia, that is, increased level of WBC. Sodium chloride, sodium 24. It is prepared from sodium 23 and emits beta and gamma rays. Use diagnostic aid. Find the efficiency of enteric coating tablets. Solution of sodium-24, filled in a coated tablet or capsule. The appearance of sodium-24 in plasma measures the efficiency, determine time and the location of the disintegration of the tablet. Applications of specific radioisotopes. Iodine-125 emits low-energy radiation than iodine-131. Iodine-125 decomposed to tellurium-125 and emits X-rays and gamma rays. Chromium-51 used in study orbital volume and survival time, protein loss from GIT, protein losing enteropathy, and absorption. Technetium-99 M refers to metastable, its half-life of 6 hours, and emits gamma rays, serves as ideal diagnostic agent. The technetium-99M is bonded to the ligand, and the resulting complex is tailored to concentrate in a specific part of the body. This provides high-quality images using a gamma camera. Calcium-44 and 45 used to study bone structure and treatment of bone carcinoma. Calcium-47 used to study of calcium absorption. Cyanocobalamin, labeled with cobalt-57, used in diagnosis of pernicious anemia, vitamin B12 absorption study. Cyanocobalamin, labeled with cobalt-60, used in vitamin B12 absorption and decomposition studies. Hydrogen, as tritium and deuterium, used in the study of total body water. N of the topic of inorganic radiopharmaceuticals. You can refer these mentioned books for this topic. Pharmaceutical Chemistry, Inorganic, by G.R. Chatwal, Inorganic Pharmaceutical and Medicinal Chemistry, by J.S. Quadri. Please take a note on important study questions. Thank you.